The presentation by the representative from Xylyx Bio, Evelyn Aranda, telling us about TissueSpeck extracellular matrix substrates as a tool to investigate cancer in vitro. So, Evelyn, yeah.
Hi, Edith, can you see me?
Yes.
Okay, great. All right, so let me share the screen. Okay, can you see it?
Yes. Good.
Okay, perfect. You see the whole slide, right? Okay, so I would like to start saying thanks to the organizer for inviting us to show our recent data and also what we are doing at Xylyx and share with you our results also. So for people working in cancer, we know that the extracellular matrix is an important component when we are considering to model this tumor microenvironment in vitro. I would like to start explaining first how the normal tissue microenvironment consists of a dynamic population of cellular and non-cellular components where the extracellular matrix plays a crucial role in regulating this organ homeostasis. As you can see in the figure, within the main functions of the ECMs are functions as adhesive substrate, also provides structure, present growth factors to the receptors, stores growth factors, and also senses and transduces these mechanical signals. However, this again is the function of the extracellular matrix in normal conditions. When we move into the tumor microenvironment, we know this extracellular matrix change. And this tumor microenvironment is characterized by a disturbance of the normal tissue homeostasis and an alteration of this normal biomechanical, like for example, the tissue stiffness, and also biochemical cues in the extracellular matrix. Interestingly, most cancer driver genes are known to be specific for certain tissue and also specific per tumor type. So when we talk about the ECM composition, we have to take in consideration that this change from organ to organ. Therefore, when we talk about the human extracellular matrix, we can't talk about just one human ECM, but several. All right, so we know that tumor metastases remain the major cause of cancer-related mortality. The tumor at the primary site, as we can see here in the figure, needs to acquire certain properties to be able to metastasize or invade in a secondary site, which is usually a different organ. Within those properties are, for example, an increase of cell motility, which would allow the cells to intravasate, go into circulation, but now they will need to survive in circulation, then later to be able to extravasate and populate a secondary site. At this secondary site, cells can continue growing and form a metastasis site or a secondary tumor, or they can go into a process that is called tumor cell dormancy. At the right, we can see a table where it's showing basically a list of different cancer types with their main sites of metastasis. And if we go into details, we can see there are organs, certain organs that are repeated and are also considered in the literature as the most common organs for metastasis, that's as bone, liver, and lung. So now when we think about that and we know that the ECMs are specific per organ, how we recapitulate this in vitro? In this paper published about 2 years ago, they compare what is the native tumor microenvironment, what is known is, to be present and important in the native tumor microenvironment, and what are the most conventional in vitro cancer models offering. So within these properties, we know that in the native tumor microenvironment, we have a limited molecular transport, and also the physical structure is important, as well as the cell-stroma interaction. In the most conventional in vitro models, we know the cells are in static conditions and also most of the time on a flat substrate, and there is no consideration on cell-stroma interaction. So we have this problem of current models lack this physiological relevance. Therefore, they cannot recapitulate key features of this metastatic microenvironment, and therefore they yield to misleading results. At Xylyx Bio, we developed this that is called the Insight Metastasis Kit that incorporates this most common that I already mentioned, common secondary site of metastasis, bone, liver, and lung, normal extracellular matrix to provide this tissue-specific composition and allowing you to recapitulate these key extracellular features of the metastatic secondary site microenvironment, which would lead to more predictive results. This one we have in two different formats, which is 2D and 3D, and also is compatible with high throughput screening. So talking about the format, as I mentioned, it's specifically for 2D and 3D. We have this 2D, which is a native code. The native code is a solubilized forms of the ECM that is used especially for these 2D assays. It's a thin layer surface. It is a coding reagent, as we can shown here in this figure. And it's also offering the proteins and growth factor present in the native tissue. The second format is the 3D hydrogel, which also contains the three most common site of metastasis of the ECM, the bone, liver, and lung. But now it provides this natural structure and the biomechanics, and the cells in this format can be seated on top of the gel or within the gel to have this, to recreate this 3D microenvironment. So let me talk and show you a little bit about our biomaterial validation data. One important feature of this ECM is its specific protein profile. In these tables, we can see in details a mass spec summary of the proteins involved in the tumor growth and metastasis. We can observe a differential tissue expression of the ECM proteins, making each component of this kit unique. As you can see, bone is first, liver and lung, and if you go through down in the list, we have these collagen, proteoglycans, and glycoproteins, and you can observe and analyze the differential expression of these proteins. The graph shows the compositional range of this collagen, elastin, and glycosaminoglycans in the three ECMs, again, bone, liver, and lung. Another important feature for an in vitro platform is to provide high consistency between lots. In general, the collagens, as you can see in these tables, are highly expressed, but this range would depend on the tissue. That would, change, but most of the time the collagens are highly expressed in any of the ECMs. In B and in C, we observe this protein profile and also collagen expression or collagen levels through different lots. And as you can observe, they are high consistent between lots. And in D, you can see how the pH remained through several months, which is one of the characteristics also of the product. Some of the data that we have been obtaining with these kits of this component are related with breast cancer cells and also lung tumor cells. In this case, for the gene expression, we use cells BT549, which are triple negative cells of breast cancer cells, and we analyze the gene expression of genes that are related in breast cancer metastasis or participate in breast cancer metastasis. And we see the cells in or on the native code of bone, liver, and lung. And we also use collagen coated plates. We use plastic as the control to analyze the gene expression. And within, this data, we can observe a differential expression of the genes. I just would like to highlight that, for example, in bone, we observe high levels of this EGF receptor in these cells, and also higher levels of this gene DRANK, which is related with osteolytic bone metastasis. So providing then a differential expression of the genes when you see the cells in this bone ECM. If we analyze proliferation, we observe here the same cells were seated on plastic and also on bone, liver, and lung hydrogels now. So the cells are in a 3D environment. So they have more cell ECM interaction. And we can see here in the graph how the cells in red are the cells on plastic. They are proliferating, and the pattern is the proliferation is higher than the cells seen in the 3D, which is pretty much what everyone observed when the cells are in 3D and we compare with 2D. However, what I want to highlight is the pattern of proliferation. Even though it's similar, you can see differences between this bone, liver, and lung. So regarding this cell ECM interaction that I also mentioned at the beginning, that is important to understand how the cells mechanically interact with these bone, liver, and lung hydrogels. We observe here just the number of the mechanical rheometry data that we obtain with the hydrogels. And in this case, we resuspend these BT549 cells, so we embedded them in the hydrogels. And then we analyze the stiffness decrease in percentage, how these hydrogels numbers are changing when they are in the presence of cells. And you can see in the graph how this data support the ability of these cells to mechanically interact with their surrounding matrix, and also how the cells respond to this hydrogel differently. Cell migration and cell invasion is also important event in metastasis. In this experiment, the same cells, breast cancer cells BT549, were seated into an upper chamber of transwell, as you can see here in the schematic, while a thin gel of bone, liver, or lung was placed at the bottom well. Then we wait for 24 hours to see how these cells would migrate through these hydrogels. And we use Madrigel and also plastic as our controls. As you can see in the graph, cells that are seated in the presence of the lung hydrogels were the ones that were highly migrating, which is statistically different from also Madrigel and plastic. And also bone was the next one that is showing a higher migration in comparison with the Madrigel and plastic. An important thing to demonstrate, or an important study for any platform or in vitro platform working in cancer or cancer metastasis, is to show that we can predict a drug efficacy. So for that, we have been working with tumor models, like in this case, lung tumor models, and doing different studies with drugs that have been already demonstrated to treat, in this case, lung cancer and also breast cancer. So in lung tumor models, we evaluate the adenocarcinoma cells A549, which are here, and also jacket. And the three drugs that we studied were erlotinib, etoposide, and KPT-185. And we observed how these cells were responding to the different treatments. As we can observe in the graph, the two cell types respond differently, where the A549 cells are more resistant. Let me show you here, in pink, or like a red is these jacket cells, and the gray are the A549. And you can see how in these hydrogels, these A549 cells and the jacket cells are responding to the drug differently. And in plastic, though, you can observe this highly different response. They are more similar. And also we assume is because here in plastic, they lack this cell ECM or cell stroma interaction. For the drug testing in breast cancer cells, we use two cell types. BT549, as I mentioned earlier, is triple negative cells, and also T47D cells, which are estrogen and progesterone receptor positive. And we use paclitaxel to treat them for 48 hours. And we analyze the percentage of viability of these cells. And as you can observe in the graph, without going into details about the behavior of these cells and the level of the receptors, how the cells respond is completely the opposite. As you can see, the cells in the viability is, in this case, they don't respond, the triple negative, for the treatment when they are seated in the bone ECM, which is not the case of the T47D cells. And this is demonstrated that the cells are correlating what is already observed in humans, that the triple negative cells that are already metastasized into bone, most of the times are highly resistant and really hard to treat. One of our also studies with these T40D cells were using tamoxifen, and these are a little longer treatment. We use 72 hours. And I just want to highlight here that we also analyze the percentage of viability. And you can observe clearly in this graph how the cells respond differently according to the ECM that are seated in. So the cells will respond to the drug according to the environment they are in. So being, in this case, again, more resistant where they are in the bone ECM, which is just probably reaching the 80% of viability. However, when they are in the liver or lung, for example, liver is this one in red, they are around the 60% of viability. So showing a high resistance in bone and a differential response on the 3 ECMs, and we also use Madrigel and plastic as the controls. This is just some data that was obtained by one of our collaborators. If you want to go more into details, this is the reference, was recently published in the summer of this year. And this is a colony information assay. They use MCF7 cells, which are the main drivers of the estrogen receptor biology source. But in this case, they contain a mutation in the estrogen receptor. That is found only in metastasis, being then a good model to investigate the behavior of these breast cancer metastatic cells. Madrigel was also used as the standard reference, and we observed how 4-hydroxy tamoxifen, which is those 4-HT, was effective in reducing the colony number in the Madrigel. As you can see, I'm not sure if it's too little for you. However, they show more resistance when they are in the liver and lung matrices. So again, all this data is supporting the fact like the cells will respond differently according to the environment they are seated in, and also they will respond different according to the drug and if they are, like organ specific, the metastasis organ specific. So I want to highlight that and saying why then the metastasis kit is important. It will be more biologically relevant. It would be useful for the standardized experiment. It's also more clinically translatable and will give you more accurate results. Please don't hesitate to contact us at info@xylyxbio.com or just visit our website, which is xylyxbio.com. Thank you.
Thank you very much, Evelyn. The floor is open for questions. Yes. Barbara asks, she says, great talk. Do you have brain-specific ECM?
Brain? Not yet. We have the list in our website. They can take a look. I noticed that I have a lot of people working in pancreas in this conference. We don't have pancreas yet and brain. We are working on that, but we don't have it available right now.
Ira?
Yeah, Evelyn, hi.
Hey, Ira, nice to see you.
Yeah, hi. I have asked you also about the brain ECMs, and we discussed this problem already. And yeah, ECMs from lymph nodes could be quite interesting for us as well. And additionally, recently I have received the message from your company, and it was written that you have fibrotic ECMs from lung and liver. But I'm understanding that these ECMs are created mostly for fibrotic diseases, not for cancer. But do you believe that it could be possible to use this also in cancer research as well?
Oh, Ira, that's a great question. Why? There are many actually cancer types that they have a fibrotic environment, like specifically like in lung and liver, but also other types. So all these features of fibrosis now are more and more related with the development of cancer. So that would be a great actually tool to use in the cancer field. We haven't yet started in-house with those ECMs. We are planning to because of the pandemic, it's been taking a little longer, but we have the ECMs, and will be the idea to explore how this fibrotic environment, tumor specific, organ specific, would be related to cancer development. Yes.
Yeah, that's a great interest of us, because we have observed some fibrotic changes in more aggressive tumors, breast cancer, and therefore this idea came, of course, but I wanted to know your opinion, whether it could be possible or not. But if you believe that this is a chance, at least to try, then we will try.
Yeah, I didn't want to include it today in the talk, but we have available fibrotic ECMs from liver and lung right now. So it's something that, more and more customers have been asking to work in, the customers that work in fibrosis, but also we have been talking with people that work in cancer, and they are, but these two fields are kind of crossing. So it would be interesting to know the role of this fibrotic environment in also in development of metastasis and in the primary tumor site. Yes.
Okay, thank you very much.
Thank you, Ira.
Any other questions? Okay.
Thank you, Adi.
Thank you very much. I think that we don't have a Dong on the line, right? So this ends the excellent sessions of today. Thank you very much to all speakers. Yeah, do you want to say anything to conclude the day?
Yes, today we have all presentations, and all of them were quite interesting. And tomorrow I would like to remind you that we will have a poster session, and therefore all poster presenters are invited to be available during the poster session. And all of you have received the link to the virtual poster session. You can just click on this link and you will enter the platform. And all posters are displayed, and all posters are provided with a button where you can press and you could enter the Zoom link specifically to the poster presenter. Not all poster presenters will be collected in one Zoom room, but all poster presenters will have their own rooms, and you could contact them at any time using this virtual poster platform. And I would like to remind you that posters will be evaluated by 3 jury members, and the best poster presenter will be awarded as usual. But as usual, not like we have done it during our previous conferences, but they will receive at least award, financial award. 300 euro is not too much, but since this conference is fully free of charge for everybody, Frontiers in Oncology provided us with some money, and therefore we would like to congratulate the poster winner with such small award. Therefore, please participate in the poster session as well. All the conference participants are welcome and are invited to participate in this poster. And of course, jury members will evaluate the posters, and poster presenters should be available. Thank you very much. And tomorrow at 9 o'clock, we will meet again. And by the end of the day, the poster session will be organized. Okay.
Thank you very much.
Thanks.
Bye. See you all.
Bye. See you tomorrow.